So today we are going to study about our chapter 1, which is cells. So let's move on. So now cytology is the study of all aspects of a cell. As our understanding of the cell has increased, so has our ability to understand all forms of life, including diseases that occur on Earth. However, there is still much work to be done in order to solve all the mysteries of the cell. Biological research laboratories all over the world are very active in this area, whether organisms are extremely small or extremely large. It is vital we understand their smallest functional units. These functional units are known as cells. Organisms range in size from a single cell to trillions of cells. To understand better all organisms around us, we must study their cells. In order to know better about every organism, we first have to study about their cells. In this chapter, we will begin with a look at cell theory. After cell theory, we will learn about the differences between prokaryotic and eukaryotic cells. So, a detailed explanation of cell parts and their functions will then follow. As much attention today is given to cancer, which seems to occur in most organisms and involves abnormal cell reproduction. We will focus on normal cell reproduction. Some time will also be spent on understanding how most complex cells may have come into existence on our planet. So this is an artist's impression of human nerve cell. And look at the picture on the right. Human nerve cell neurons, essential to our lives. Because of these cells, we are able to acknowledge and remember everything and respond to our surrounding. Neurons are usually very efficient, but sometimes things go wrong. Can we gain a greater understanding and better treatment of conditions such as depression by learning more about how these cells functions? So our first unit is 1.1, cell theory, cell specialization, and cell replacement. So now let's read the understanding. According to the cell theory, living organisms are composed of cells. Organisms consisting of only one cell carry out all the functions of the life in that cell. Surface area to volume ratio is important in the limitation of the cell size. Multicellular organisms have properties that emerge from interaction of their cellular components. Specialized tissues can develop by cell differentiation in multicellular organisms. Differentiation involves the expression of some genes and not other in the cell's genome. The capacity of stem cells to divide and differentiate along different pathways is necessary in embryonic development and also make the stem cell suitable for therapeutic uses. So, according to cell theory, living organisms are composed of cells. And try to remember all these points. So, organism carry out only one concern of only one cell carry out all the function of life in that cell. So we will learn. We are going to read all this in this chapter. So application skill, application questioning the cell theory using typical example including striated muscles, giant algae, aseptate fungal hyphae. Application Next application is investigation of function of the life in paramecium and one named photosynthetic unicellular organism, and use of stem cell to treat Stargardt's disease and one another named condition. Ethics of the therapeutic use of stem cells from the specially created embryos from the umbilical cord blood of the newborn baby and from adults own tissue. And the skill is like use of light microscope to investigate the structure of cell and tissues, and drawing of cell calculation, the magnification of drawing and actual size of structure and ultrastructures showing in drawings or micrographs. So now there is some guidance. The students are expected to be able to name and briefly explain these functions of life: nutrition, metabolism, growth, response, excretion, homeostasis, and homeostasis and reproduction. Chlorella or Cynesmus are suitable photosynthetic unicells, but Euglena should be avoided as they can feed heterotropically. Scale bars are useful as a way of indicating actual size in the drawings and micrographs. So now let's read. Cell theory. It has taken several hundred years of the research to formulate the cell theory that is used today. Now, many scientists have contributed to developing the three main principles of this theory. These three principles are all organisms are composed of one or more cells. Cells are the smallest unit of life, and all cells come from the pre-existing cells. So these three principles we have to learn. All organisms are composed of one or more cells. Cells are the smallest unit of life, and all cells come from the pre-existing cells. And now cell theory has very solid foundation, largely because of the use of the microscope. Robert Hooke first described cells in 1665 after looking at the cork with self-built microscope. A few years later, Antony van Leeuwenhoek observed his first living cells and referred to them as animalcules, meaning little animals. In 1838, the botanist Matthias Scheleden stated that plants are made up of independent separate beings called cells. 1 year later, Theodore Schwann made a similar statement about animals. So this whole cell theory was taking place there, which started in 1665 by Robert Hooke, who first described cells. And he just started it by looking at the cork with self-built microscope. So, and after that, till Theodore Schwann, who made similar statements about animals after looking at Matthias Sladen. So this whole cell theory on which biology depends. So the second principle continues to gain support today because so far no one has been able to find any living entity that is not made up of at least one cell. Some very famous scientists such as Louis Pasteur in 1880s have performed experiments to support the third principle after sterilizing chicken broth soup by boiling it. Pasteur showed that living organisms would not spontaneously reappear. Only after exposure to pre-existing cell was life able to re-establish itself in the sterilized chicken broth. So now cell theory, we have read all these discoveries, and some very famous scientists like Louis Pasteur in 1880s have performed experiments to support the third principle. Like this was the second principle, and here the first principle here. The third principle and the most famous was like supported by Louis Pasteur, who was the most famous scientist after sterilizing chicken broth by boiling it. And yes, so there is a nature of science here. As with the most scientific theories, cell theory is not without areas of concern and problems. A key characteristic of the good scientist is skeptical attitude towards theoretical claims. To overcome and validate this skepticism, evidence obtained by observation or experimentation is essential whenever possible in science. Controlled experiments are needed to verify or refute theories. These experiments have a control group and variable groups. The group are kept under similar condition apart from the factor that is being tested and questioned. The factor being tested is referred to as the independent variable. The dependent factor is measured or described using quantitative or qualitative data. This is just the nature of science, and just telling about that this theory. Relatively recent findings that have raised questions about cell theory include observation of striated muscles, giant algae, and acceptate fungal hyphae. So as this chapter develops and more information about basic character of cell is learned, some recent findings will be discussed. Okay, so now here comes functions of life. All organisms exist in either a unicellular or multicellular form. Interestingly, all organisms, whether unicellular or multicellular, carry out all the function of life. Now, there are two types of cellular organism, like the unicellular and multicellular. So these organisms carry out functions whether they are unicellular or multicellular. So these functions are metabolism, reproduction, homeostasis, excretion, growth, response, and nutrition. Every organism grows. It shows grows because it's living, as it's living, and shows response, and nutrition is also important for them to live, and metabolism takes place in their body, like the digestion and all that. So now all these functions act together to produce a viable living unit. Metabolism includes all the chemical reactions that occur within an organism. So cells have the ability to convert energy from one form to another. Growth may be limited but is always evident in one way or another. Reproduction involves hereditary molecules that can be passed to offspring. Responses to stimuli in the environment are imperative for the survival of organism. These responses allow an organism to adapt to the environment. Homeostasis refers to the maintenance of the constant internal environment. For example, an organism may have to control fluctuating temperature and the acid base levels to create constant internal environment. Providing a source of compounds with many chemical bonds that can then be broken down to provide an organism with the energy necessary to maintain life is the basis of nutrition. Excretion is essential to the life because it enables those chemical compounds that an organism cannot use or that may be toxic or harmful to it to be released from the organism's body. So that's what these are all about, the function of life. These are the key points, and this is their definition. Like, in order to describe them, if somebody asks you to describe these function of life, then you can just read it and learn it, so and give the answer. Now we have some highlighted points. So viruses, prions, and viroids are not considered to be living organisms. They cannot carry out the function of life on their own. However, they may use cells to perpetuate themselves. Now, the function of life manifests in different ways in different type of organism. However, all organisms maintain the same general function that allow them to continue life. You may see different terms of these function in other sources. Okay, two organisms can be used to demonstrate the functions of life: the paramecium and the chlorella. Let's read about them. Paramecium is unicellular member of the kingdom known as Protista. So it is the unicellular membrane. A member unicellular means it has just one cell, and it belongs to the kingdom known as Protista. And now study the diagram of paramecium to become familiar with the organism's basic structure. So here we have the structure of paramecium. Paramecium, this is a single cell organism, may be used to demonstrate several of the functions of the life. So this is the anterior end and this is the posterior end. Now anterior contractile vacuole, this is shown by this. The posterior contractile vacuole, this is shown by this. And the oral groove, this hole is the oral groove here. And macronucleus is this bigger part and micronucleus is the smaller part. The trichocyst is this part, and the cilia are these thread-like structures, and food vacuole are these smaller circles, and cytostome is the part displayed here, and the anal pore, this is like here to excrete the waste out of the structure, and the buccal cavity with the rows of cilia used in feeding, like to take in food. So, and the oral vestibule here, these all have some of the other functions. Now Let's read permission and the functions of life. Safety alerts: be cautious of the sharp objects. Only use a 9-volt battery as an electrical source. Make sure your instructor checks your setup before you begin. Wash your hands thoroughly and soap and water before and after the procedure. So now permission can be used to demonstrate the functions of life in several ways. Place a number of permission into the Syracuse dish or an evaporating dish with the positive and negative electrodes of the low voltage electrical charge on the opposite side. A simple 9-volt battery will usually trigger a response. Do not use electricity of higher voltage and otherwise the organism will be harmed. Low voltage electricity can apply for several minutes. The dish should be placed on the stage of the dissecting microscope. A strong magnifying lens may also be used. Describe the movement and the final location of the large proportion of the paramecia. Once this activity has ended, remove the electrodes and add several small but visible pieces of hard-boiled egg yolk. Again, using the magnifying instrument, make observation of the movement and the final location of the paramecia. Finally, to a culture of paramecia, add a drop of very dilute acetic acid, which is vinegar. Once again, report on the movement and the final location of the paramecia. When you have finished these tests, your teacher will explain what should be done with the organisms. Respect for life is very important in our studies, and the policy on animal experimentation must be followed at all times. Using what you know about the function of life, explain why the paramecia moved in the ways you observed. So, okay, this was an experiment. Now, the next organism we will look at is Corella. The compared with Paramecium, Corella has completely different approach to the nutrition. Corella is single-celled organism that has one very large structure called chloroplast inside a cell wall. The structure enables the conversion of the energy in the sunlight to a chemical energy from form called carbohydrate. So the next organism we'll take is Corella, and which compared in comparison to Paramecium, Corella has completely different approach to nutrition. Okay, and Corella is single cell organism. Paramecium was also single cell organism, and Corella is also single cell organism. So that has one very large structure called chloroplast inside the cell wall. Corella. This structure enables conversion of energy in sunlight to chemical energy. So they what happens is they absorb sunlight and this chloroplast inside the cell wall converts the sunlight into chemical energy uh, called carbohydrate. The carbohydrate provides the major nutritional source for the organism, and let's study the diagram of the chlorella. Now this is a diagram of chlorella. If we have a cell membrane here and a cell wall which is shown in blue color, a nucleus here and cytoplasm, this area, and the chloroplast here, which converts the sunlight energy into chemical energy in the form of carbohydrate. Now, a corella, a common freshwater organism, this organism has been used by many researchers to determine the details of factors that affect the process known as photosynthesis. The structure labeled chloroplast is especially important in this process. So now let's read the nature of science. Perhaps in the design of Perla activity, you had an idea based on your previous experiences in the science about what the outcomes of the procedure would be. This idea is referred to as hypothesis. Scientists from hypothesis that can be tested by observation and other experimentation. These tested hypotheses may ultimately serve to simply and unify existing scientific ideas. Controlled experiments are the best way to investigate the relationships between the two factors or the variables. However, this type of experiment is not always possible. In this case, statistical analysis of the data may indicate the correlation. As time and research proceeds, a causal relationship may be seen. Objective data, both qualitative and quantitative, are used to establish relationships whenever possible. It is essential to repeated measurement are taken and that large numbers of reading are taken so that the data collection is reliable. The scientists spend a lot of time working with the people from other disciplines in order to gain a greater understanding of their findings. They also read current scientific articles throughout their career in order to gain further insight into the research. Eventually, a researcher may decide to publish his or her findings on the appropriate scientific journal. For this to happen, an article undergoes a peer review process, which means several scientists working in the same field read the article before it is published to make sure the methodologies and findings are sound and honest. So whole thing is done for this. So now cell and size, let's read about that. Cells are made up of number of different subunits. So cells are the smallest unit of life, but they are even made up of number of different subunits. These subunits are often of the particular size, but all are microscopically small. They microscopically small means they are very small. We can only see them by microscope. So in most cases, the use of microscope with high magnification and resolution are needed to observe cells and especially their subunits. So resolution refers to the clarity of the viewed object. Light microscope use light passing through the living or dead specimen to form an image. Stains may be used to make it easier to see and detail. Electron microscope uses electrons passing through the dead specimen to form an image and provides us with the greatest magnification over this image and the resolution. Now there is a comparison between the light and the electron microscope. Let's see. So light microscope we have here and electron microscope we have here. Light microscope is inexpensive to purchase and operate, so it is very cheap and it looks good. An electron microscope is expensive to purchase and operate. An light microscope is simple and easy specimen preparation. Complex and lengthy specimen preparation is with electron microscope. And light microscope magnifies up to 2000 times, and electron microscope magnifies to over this much huge quantity. So now light microscope specimens may be living or dead, and electron microscope specimens are dead and must be fixed in a plastic material. They should always be fixed in plastic material, and specimens are usually dead. So cells and their subunits are so small they are hard to visualize. It is important to appreciate their relative sizes. Cells are relatively large and then is in decreasing order of size are organs, bacteria, viruses, membranes, molecules. So the decreasing order of size is the organelles, bacteria, viruses, and then membranes are smaller than viruses, and molecules are much smaller than membranes. So if you want to calculate the actual size of the specimen seen with the microscope, you need to know the diameter of the microscope's field of vision. This can be calculated with the special micrometer or a light microscope with the simple ruler. The size of the specimen can then be worked out. The drawings or the photographs of the specimens are often enlarged. To calculate the magnification, we have a formula here which is size of the image and over the size of the specimen. We have to divide them and then we get the magnification. Now the scale bars are often used with a micrograph or drawing so that the actual size can be determined. Scale bars and magnification will be addressed in more detail in the later practical activity. Now we have a worked example here. Let's read. The length of the image you are looking at is 50 millimeters. If the actual length of the subject of the image is 5 micrometer, what is the micro? It has mu m and it means 5 micrometer. What is the magnification of the image you are looking at? So to find magnification we have to simply divide the size of the image by the size of specimen. So magnification is 50 millimeters over 5 micrometers. So it will be like, if we convert them to micrometers, it will be like 50,000 over 5, and then it's 10,000 times. So magnification can also be found out by this method. 50 mm over 5 micrometer will, which would be then 50 into 10 raised power minus 3 meters divided by 1 into 10 raised power minus 6 meters, which is 10,000 times. Now, limiting the cell size. So the cell is the smallest object. You may wonder why cells do not grow to larger sizes, especially as growth is one of the function of life. Because humans grow, human beings grow, their height grows, the even the nails grows, the hair grows, and eventually the size of human beings, they grow, and every living thing, it grows. So why don't the cell grows? This is the question here. So there is a principle called the surface area to the volume ratio that effectively limits the size of the cell. In the cell, the rate of the heat and the waste production, the rate of the resources consumption are the function of that depend on its volume. So the rate of heat, the waste production, and the rate of resource consumption are the function of that depends on its volume. The most of the chemical reactions of the life occur inside a cell, and the size of the cell affects the rate of those reactions. The surface of the cell, the membrane, controls the what materials move in and out of the cell. A cell with more surface area per unit volume, the cell which will have the more surface area per unit volume, is able to move more materials in and out of the cell. So it will be easy for the cell for each unit volume of the cell. As the width of an object such as a cell increases, the surface area also increases, but at a much slower rate than its volume. Now, the width of the object such as cell increases, then the surface area also increases, but at a much slower rate than the volume. So this is shown in the following table. Like, the volume increases by a factor calculated by the by cubing the radius. At the same time, the surface area increases by a factor calculated by squaring the radius. Now, if we look at the table, it is like the volume increased by a factor calculated by the cubing the radius. If we like cubing the radius is like raised to power 3, and the same time surface area increased by factor calculated by squaring the radius. So volume can be like cubing the radius and surface area by squaring the radius. So now let's look at the table. Factor cell radius is 0.25 and measurement is 0.50, and then 1.25. Then surface area we have 0.75 and 3.14, 19.63. Volume is 0.07, 0.52, and then 8.18. And surface area to volume ratio is 11.29 ratio 1, 6.04 ratio 1, and 2.40 ratio 1. Now, sphere formulas. Surface area equals 4 pi and radius is squared, and then volume is 4/3 pi. These are the sphere formulas. And if you want, you can learn these formulas. And 4/3 pi to the cube of the radius. Then now this means that a large cell compared with a small cell has relatively less surface area to bring in materials that are needed and to get rid of the waste. Because of this, cells are limited to in the size they can reach and still be able to carry out the function of life. Thus, large animals do not have larger cells, instead of they have more cells. So they don't have a large number, like a bigger size of the cell. They have the bigger number of cells. Okay, so we have more number of cells than a squirrel or a rat or even a dog. Okay, so cells that are larger in size have modification that allow them to function efficiently. They have larger number of size, have modification that allow them to function efficiently. This is accomplished with changes in the shape, such as being long and thin rather than spherical. Every cell is not spherical. Like the blood cell, it is like somewhat similar to a donut. Like in between, it is pressed like this. Maybe you can see the blood cell. So now this is accomplished with changes in the shape, such as being long and thin rather than spherical. Some larger cells also have infoldings and outfoldings to increase their surface area relative to their volume. So cell can be of every type. Like here we have this is the computer artwork of the egg cell fertilized during vitro fertilization and now undergoing the first cell division. So the you can easily see the cell division here. These are the two nucleus and the cells are dividing. So let's read cell reproduction and differentiation. One of the function that many cells have is the ability to reproduce themselves. Cells have the ability to reproduce themselves. Remember viruses just divide, they just divide very fast, and that's the reason why viruses spread with a much faster rate. Okay, so in multicellular organism this allows growth to happen. It also means damaged or dead cells can be replaced. Multicellular organisms usually start their existence as a single cell after some type of sexual reproduction. This cell This single cell has the ability to reproduce at a very rapid rate, and resulting cells then can go through a differentiation process to produce all the required cell types that are necessary of well-being of the organism. The number of different cell types that can rise from the one original cell can be staggering. The differentiation process is the result of expression of certain specific genes but not other. Now, genes, segments of DNA on a chromosome. Genes are the segments of DNA on a chromosome. Genes are fine, are like we can look at genes in the chromosome. Enable the production of all the different cells in the organism. Therefore, each cell contains all the genetic information. Each cell there has all the genetic information of the organism needed for the production of the complete organism, complete new organism. However, each cell it will become specific type of cell depending on which DNA segment becomes active. So here we have some point. Cancer cells are example of cells that undergo extremely rapid reproduction with the very little or improper differentiation. So they undergo extremely rapid reproduction with little or improper differentiation. The result is a mass of cells like a tumor with no useful function to the organism. So now some of the cells have a greatly reduced ability to reproduce once they become specialized or lose the ability altogether. Nerve and the muscle cells are good example of this type of cell. Other cells, including the epithelial cells, such as the skin, we have the skin cells, retain the ability to reproduce rapidly throughout their life. The offspring of these rapidly reproducing cells will then differentiate into same cell type as the parent. And now one of the result of the cell production and the subsequent differentiation process that occurs in multicellular organism is emergent properties. These properties depend on the interactions between all the different parts of the particular biological unit, such as the cell. When you look at the function of each part of the cell, it is less than the overall function of the complete cell. In other words, the whole is more than the sum of its parts. To continue with the emergent concept, a whole multicellular organism is capable of carrying out more function than the sum of the functions that each cell is specialized in. The ultimate example of the emergence is the collection of inert non-living molecules that is capable when functioning together or creating a living entity that demonstrates the functions of life. So now we have stem cells here. Stem cell. There are populations of cell within organism that retain their ability to divide and differentiate into various cell types. These cells are called stem cells. So stem cells are what population of cells within organism that they have retained their ability to divide and differentiate into various cell types. So these are the stem cells, and plants contain such cells in regions of the meristematic tissue. Plants have the stem cells in the regions where they have meristematic tissue. Meristematic tissues occur near the root and the stem tips. So we can imagine and see at the stem tips they have the stem cells there, and now and are composed of rapidly reproducing cells that produce new cells capable of becoming various type of tissues within the root or the stem. Like the stem grows at a much faster rate, and roots also grows at much faster rate in plants, so they have the stem cells there in the meristematic tissue. And gardeners take advantage of the cells when they take cuttings from the stems of the roots and use them to propagate new plants. Gardeners, they nowadays they may take a good advantage of these cells because they can propagate a larger number of plants. And by this technique we have a hydroponic technique, like they take the cuttings, maybe like some of the vegetables and other plants, and they put them in water and they grow in the greenhouse. And even the countries with receive low sunlight during the year, they grow vegetables and fruits, and like other cuttings, so in the greenhouse, and they get extremely amazing results, and by that they earn a greater profit. So that's good technology. In the early 1980s, scientists found pluripotent or the embryonic stem cells in the mice. In the mice, you know what is mice. These stem cells retain the ability to form any type of cell in the organism and can even form a complete organism. When stem cells divide to form specific type of tissue, they also produce some daughter cells that stay as stem cells. This enables a continual production of a particular type of tissue. Medical scientists show the possibilities of using such cells so as to treat certain human diseases. However, one problem discovered early on in the stem cell research was that stem cells cannot be distinguished by their appearance. They can only be isolated from other cells on the basis of their behavior. So stem cells can only be isolated from other cells on the basis of their behavior. They cannot be distinguished by their appearance. So stem cells research and treatments. Recently, some very promising research has been directed towards growing large number of embryonic stem cells in the culture so that they can use to replace differentiated cells lost as a result of injury and disease. This involves therapeutic cloning, Parkinson's, and Alzheimer's disease are caused by the loss of proper functioning brain cells. If like a person loses the brain cells, and it is hoped that the implanting stem cells could replace many of these lost or the defective brain cell, thus relieving the symptoms of disease. That's really good, like using the stem cells to replace the defective brain cells. And with some forms of diabetes, the pancreas, with some forms of diabetes, the pancreas is depleted of essential cells, and it is hoped that a stem cell implant is that organism could have positive effects if they have stem cell implant. As at present, most of the research on the stem cell is being carried out using mice, using the rats. It will probably be some time because before this approach treatment because widespread in humans. We have a fact here. In 2015, stem cells were used successfully to help restore the lost insulation of the nerve cell in rats, thus resulting in greater mobility of this in these animals. This is all about our technology. Okay, so stem cells are being utilized in a number of ways by scientists around the world. One area of research involves using human embryonic stem cells in order to understand humans' development better. This research involves studies of the cell division and differentiation. Other scientists are using stem cells to test the safety and effects of the new drugs. The information in the area is essential to understanding of how these drugs might affect the differentiating cells in existing organism. Another way interesting area of the study involves cell-based therapies, especially as they may have a positive influence on the treatment of disease, the trauma such as the Alzheimer's disease and the spinal cord injuries, the heart disease and the diabetes and the burns and strokes that people have. So, however, there is a type of stem cell treating that has been used successfully in humans for many years, as well as pluripotent stem cells. There are tissue-specific stem cells. These stem cells reside in certain tissue types and can only produce new cells of that particular tissue. They can only reside in certain type of tissue types. For example, the blood stem cells have been introduced routinely into human to replace the damaged bone marrow of some leukemia patients. The blood stem cells have been introduced routinely into the humans to replace the damaged bone marrow of some leukemia patients. Stargardt's disease is an example of the human condition that is in the early stage of the being treated with the stem cells. Stargardt's disease is an inherited disease because it's caused by both parents passing on the gene of the offspring that causes or defect in the processing of the vitamin A. They have defect of the processing of vitamin A. Vitamin A is essential for the light sensitive cells in the retina. Retina is like in our eyes to function properly. Vitamin A is essential for the light sensitive cells that sense lights. Remember, retina sense lights in the eyes. So, with Stargardt's disease, within the first 20 years of the patient's life, he or she begins to lose his or her central vision. So, he or she, like the person in 20 years, can lose his complete central vision. Later on, peripheral vision loss occurs, and which eventually leads to blindness. So, it leads to blindness. The person cannot even see. In March 2010, a stem cell treatment was begun that was designed to protect and regenerate photoreceptors in the retina that are damaged by Stargardt's disease. Currently, the particular stem cells being used for the treatment in humans are human embryonic stem cells. The study is ongoing, but early results are promising. There has been much sharing of data involving stem cell research. However, many nations have banned or restricted research in this area because of local cultural and religious traditions. There are ethical issues involved in the stem cell research. The use of pluripotent stem cell is particularly controversial. These cells are obtained from embryos, largely from laboratories carrying out in vitro fertilization (IVF). Harvesting these cells involves the death of an embryo, and some people argue that this is taking a human life. Others argue that this research could result in the significant reduction in human suffering and is therefore totally acceptable. So that's all for today. And here are some exercises we have. You can practice these questions on your own. If you want, you can give me the solution on the comment or just practice. These are some really nice questions that regarding what we have read so far. So that's all for today, and bye bye and have a nice day.